Well, I'm hoping that my little photo of our new baby with the giant brain and four hands got your attention. Um, Miriam and I are probably too old to have such a baby, but I didn't want to talk. I wanted to talk about some things which I think everyone should know about something that happened in 2012, which could affect everyone in the long term. Now, I mean, their history has lots of ups and downs, and certainly Russia going into the Ukraine is a worry that we all have today. But I want, and there's pluses and minuses and directions and changes sideways and in our political history, which I think kind of even out over time. But underneath all that, there's another history, a history that happens quietly and that most of us don't recognize when it's happening. And it's what I call a scientific history. It's really actually one reason I became a scientist. And there's some things that happened in 2012 that I think everyone should know about. Now, if you're a biologist or maybe a medical person or extremely well-read, you probably know all this already. But I wanted to sketch it out for the average person in a way that's easier to understand, because I think it's something that is important to all of us. Now, you might say at the beginning, or think about it, at least at the beginning, is it a good thing for people to be able to do genetic engineering? Is that something which is inherently evil or bad, or is it something which has tremendous positive things about it? I think both are true, and we'll get back to that and talk about it more as I go along. But first, I thought I'd like to explain in a straightforward way what happened in 2011 and 2012.
Google, hello.
Well, let's start with the basics of biology. And maybe some of this is obvious, but the human body has about 37 trillion cells in it. Of course, it starts off with just one, and then it starts dividing. The cells are clearly very tiny, and as you can see, each cell has DNA in the nucleus, and the DNA is basically the genetic code that tells the body how to be born, how to grow, how to reproduce itself, how to die. The DNA encodes things called messenger RNA, which come out of the nucleus, and they go into little molecular factories called ribosomes, and the ribosomes take the instructions and they pop out proteins, which are really the worker bees of the cell. That it's the little molecular things which carry materials around in the cell. They build the outer walls and everything. I've drawn this with the cell on the outer edge because one thing I always wondered about was, well, if you start with just one cell and it keeps duplicating, and those are stem cells at the beginning, and how does it decide to make a heart or a muscle or other types of cells? Well, it starts very simply. The cells that are on the outer surface sense that there's a chemical difference there. There's something missing on the outside and they say, this must mean we're supposed to become skin cells. And so they start to change. And so the outer surface starts to evolve into something which becomes skin. The other thing that's really fascinating is during this process, a hole forms in the middle of the ball, and which eventually becomes the gastrointestinal tract, the mouth and everything else along the way. So once it starts this process of developing, and new cells are made all the time, they start to develop to do the specific jobs that they're supposed to do. So that's kind of the basics of how a cell works. There's actually thousands of things going on, even in one cell. And it's really a fascinating subject. Now, I want to go into a little bit more into how this works. So the next slide shows you a picture of how the DNA works. As you must all have heard, that DNA is sort of a double helix where there's 4 possible chemicals along the string, and they're kind of in pairs with the complementary thing on the other side of the double helix. But the four things, T, G, C, and A, basically have the instructions for building a human body or a plant or anything else. For humans, there's separate chromosomes, but all the DNA, if you had added up, would be about 5 feet long, and it has about 3 billion pairs, which are into this genetic code. Now, people talk about genes and genetics all the time, and so I wanted to explain that a little bit better. There are segments along the DNA code which are genes. They're the instructions for building a protein or something else out in the cell in general. Now, when you can tell where a gene is because at the ends of it, there's some sequence of instructions which say this is the end, where this is beyond this gene. So you should stop here. What happens is that things are moving so quickly inside of a cell that all sorts of molecules bump into the A, G, C's, and T's along the edge. And when things are complementary, they say, oh, I can latch on here. And what happens is that you get sort of a complementary messenger RNA, which kind of forms along there. And when it gets to the stop parts, it says, oh, we're done now. And so as soon as that happens, it kind of peels off and goes out and leaves the nucleus and starts making the proteins with the ribosomes and so on. Now, this is the thing that was used to make the coronavirus vaccine. People have been looking at how to deal with coronaviruses for several years. So they had a general way of doing that. But I think, and we all tend to think that it took, from January or so in 2020, I guess, when the virus first started appearing generally, to October, November, before it started being available, that it took all that time to make the virus, the vaccine. But actually, it only took about a month. People assembled this mRNA really quickly. They kind of had the parts they needed, so they put it together and they tested it against the COVID-19 virus. And very quickly they said, this works. This seems to be doing, preventing the little spiky things on the coronavirus from infecting other cells. And so they started trying it out on a few people, and then on hundreds, and then on 10,000. And that's what took the 9 months to make sure it was working and it wasn't hurting other things.
So that's basically how they built the coronavirus vaccine, which I hope you've all had.
I'd like to tell you a little bit of the story about how they figured out this gene editing capability. And there was people recognized that although mammals and bats and people and everything can get sick from viruses, they noticed that bacteria have been around for 3 billion years or so on Earth, never seem to be bothered by viruses. And so people started studying bacteria and looked at, well, what are they doing to protect themselves from viruses? Because there's no reason why they shouldn't be affected the same way everything else is. And what they found was that there were sequences in the DNA for, or the RNA for bacteria, which had these kind of long repeating sequences where the same thing kept coming up over and over and over again. The yellow parts were not repeating, but the green parts, let's say, were. And then at the end of that sequence, they had an enzyme called Cas9. There's other enzymes too with different names, but what they found was that this sequence of repeating things, which I might as well jump to the bottom line here, are called clustered regularly short palindromic repeats, or CRISPR, which you may have heard the term. And those those sequences were able to, whenever a virus would get into a bacteria, this thing would notice that, and it would put the code for the virus into the yellow parts. And then what happens is that this little sequence would go up to the DNA of the bacteria, and it would cut out anytime the virus got into the bacteria's DNA. This thing would cut out the part that was for the virus, so the virus could never duplicate and never take over the cell. So, like I say, back in around 2011, people discovered this, and they figured out that's how bacteria were protecting themselves. And it took about another 9 months, because everybody was trying to do it, to prove that this actually could work in humans. And so there were several parts, the Cas9 enzyme and the CRISPR part, and then there's another part called tracer. And what they discovered was that the tracer part could aim it towards any part of DNA and say, this is the section I'd like to remove. And when it was removed, so if it was like a bad gene or one that had mutated and was causing medical problems, it could just cut it right out. So that's basically how CRISPR works. And it's an incredibly powerful tool because it can basically edit any part of the human DNA that you want it to. And so over the last, since 2012, 10 years almost, people have been experimenting with this. And so I think it's important to understand what the decisions were about how this technology could be used. In a nutshell, we can now redesign any plant, animal, or humans themselves. It's not trivial, but it's not really hard either. This can be done. And there's been many conferences and gatherings of scientists and others to decide how to deal with this new technology. And kind of generally speaking, they've come up with these rules. First of all, we need to focus this technology mainly only on living persons. That is, it's a somatic thing, it's called. What we don't want to do is make changes to sperm and egg cells of humans that would change the germline, the DNA of humanity forever, because it would be something that could be reproduced. So people have stayed away from that, except for one Chinese guy who actually did something to limit the AIDS virus in some babies, and some children were born who are now immune to HIV.
He was roundly condemned for this. I
think the other thing safe to say is people are focusing on simple changes. one gene changes where something has gone wrong, and I'll talk about some of them in a minute. If you talk about something like intelligence or getting 4 hands on a baby, those are things which require multiple gene changes, and they're probably many, many years off. But they can be done. It's not limited too much. The other thing they've kind of decided is they don't want to work on defects, the medical issues and other things, rather than strictly, trying to enhance people. For example, changing eye color or making longer arms or making babies 7 feet tall, or not babies, but when they grow up. And those kind of things are possible. In fact, there's experiments were done with a frog where they did a CRISPR change, and they put in a new sequence which makes their muscles like 5 times bigger. So these frogs were able to like super jump and go around. So you can do enhancements, but most scientists are trying to stay away from that. The other thing is that this is a really powerful technology, and so I think a general thought is that we should do it slowly, and we're not ready for any really big changes. But I think it's also really important to understand that especially the scientists and others involved in this have been very adamant about saying we're not going to stop experimenting. This is something which is so valuable and so powerful that it needs to be understood and used when appropriate. So experiments are continuing on. So I think it's safe to say these rules are not permanent, and they certainly don't apply to plants or animals. So I think we're going to start to see a lot of genetically modified animals, and as well as not just tomatoes. But I think it's important to say that this gives us capabilities we've never had before. It's a world-changing line that we've crossed. Of course, there's been genetic enhancement going on from ancient times. That's when 2 people meet and have babies. They are, in a sense, selecting mates and partners who, generally speaking, will improve the line. And so, in a sense, this has been going on from the beginning of time. Things, as long as there was sex involved, and it's possible, you can make a choice. And I think that's going to be true as some of these genetic enhancements happen. If a genetic enhancement happens which makes a person, just undesirable, that trait's not going to be passed on very much. So I think the net effect of it is that it's sped up evolution for humans and for other things dramatically, because we now change things directly that we couldn't before. And so I've said here that it's evolution might be about 1 million times faster now than it has been. So let's talk a little bit about the implications of this. How far should we go on this redesigning of humans or anything else for that matter? As I mentioned before, one of the dimensions of this is repairing defects versus making enhancements. And I have a chart here which has kind of an overview of what some of the things we've thought of. And I've given this talk in meetings, and people have thrown in more ideas. The ones that are in red on the left are things where the genetic engineering has been tried. A young man was cured of sickle cell using this technique. Sickle cell is a nice problem to solve because it only involves one gene. So if that gene is something that's causing the cells to be sickle shaped. And that boy in particular was having trouble getting his breath, and it really makes it difficult to breathe. And on the other hand, sickle cell is sort of a preventer of malaria. And so it developed in Africa in a sense to make people immune from malaria. So there's pluses and minuses of it, but it certainly helped this young man to get the gene changed. It's helped his life. A lot of these things are discussed in much more detail in a movie called Human Nature. I think you can get it on Netflix. And it talks not only about that boy, but also about, it has much better graphics about how the genetics work, and have some of the key players who developed this technology are in the movie. It's about an hour long, so I would recommend checking that out. There are, as I mentioned, HIV, there was a person who experimented with that. There's a lot of other diseases or things which are more complicated. Diabetes, and Alzheimer's, and so on. And some of those may be things which you'll be seeing preventive technology for in the next few years, at least on the individuals who are involved. There's Huntington's disease, for example, is a horrible disease that doesn't show up until you're older. And so if you have the gene for that, you won't know until you're 40 years old. And it's like a horrible way to go. And it's also tricky because your children can pass this on by just having, one half of from one parent can be passed on to future generations. And it doesn't necessarily show up right away. It's that gene is sort of floating in there, and you don't, sometimes generations will pass and nothing will happen, and then suddenly it'll show up again. But there's certainly a slew of different illnesses and problems which might be able to help, but it becomes much more controversial when you go over to the right side here, where you talk about, I mentioned giving a frog more muscles, but you can imagine other things. I can imagine rogue regimes creating super tall people for winning at basketball or the Olympics. People with and there's dozens of things you can imagine. In my initial picture, I showed the baby with 4 hands. I think that would be useful, but maybe not, and maybe that would turn off any prospective spouse for that person. Anyway, so this, I wanted to make the point that this is one dimension, only one. This continuum from defects to enhancements, where we're going to, over time, decide how far we want to go. And these things will start showing up. There's another key question, which is who decides? Who decides if, does the individual, can they just say, hey, I want to be defect free from all the diseases? Let's say there's 20 things which we develop ways to prevent. Can an individual say, give me all that stuff? Or are some of these things are decisions that need to be made by the community itself? For example, if we were able to find 2 or 3 keys to longer life, the anti-aging one on the right of the chart, is that something which is good for humanity, that we all live to be 200 years old? Or is that something which, and should, if you're sick and you're 90 years old, you might say, hey, I want that. I want to live a much longer time. But is that the kind of decision that people should be making as individuals? The third dimension might be who gets to choose? Is this something which is free choice, or is it something which is going to be managed by government? Is government going to say certain things are allowed and certain things aren't? Who is there an overall controlling body? Right now, it's not controlled much except by the scientists themselves, who kind of making up the rules as they go. I probably can't do this without putting in a little plug for my book, where I thought about all these things. I thought about what happens if you could do genetic engineering. What would we actually do, say 50 or 100 years out from now? So I wrote a novel, sort of an adventure novel, which looked at genetic engineering. It looked at the effect of artificial intelligence, putting implants so that you could, have a phone kind of built into your head. What would happen with longer lives? And then, of course, the negative sides of these things. Bio and cyber warfare. But I think as I wrote through and wrote the book, I had to think about, well, what's the implication of this? The heroes in the book, which are pictures are shown there, were kind of having to deal with these things. They were in a position to influence the results. And so that's kind of the heart of what that book is about. And it's available kind of from all over the place. Anyway, finally, I want to just say, make sure that you understood that, and it very quietly, in around 2012, the world changed and humanity changed. And this is something which your children and your grandchildren, your great-grandchildren will have to deal with more and more. It's not the sort of thing which happens overnight. 10 years have already passed, and we don't have cures for all of things. Although, in some sense, the vaccine that was developed for the coronavirus was made possible by this. And I think it's interesting to note that like 60 or 70% of all viruses that we know about have certain characteristics in common. And so one of the things people are working on very diligently right now is to come up with a general approach for those common things, to a genetic, a vaccine which can basically prevent viruses with all those characteristics, and which would conceivably eliminate or 60 or 70% of all flus. So there may be a vaccine coming along which is much more general than the coronavirus vaccine. I think bioengineering like this is a powerful ability, and like any science, it has potential for both good and evil. Some of you might look at this and say, this is just horrible. And I asked you at the beginning to think about what you think about this. And, if you can, feel free to get back to me and give me your opinions. But my general sense is that over time, science gives humanity a new capability, and that over time that we try the bad things, we try the evil things, we try the good things. And over time, people start saying, well, let's not do that anymore. It's like nuclear weapons. We kind of said, oh, we tried that and it's really bad. So let's not do that anymore. And I think a lot of technology is like that. You might say, well, smartphones are fine, but they're eating up too much time, and it's really causing problems for our children as they grow up. And that's partly true. And yet people find ways to avoid those problems. That over a long enough time, the bad results of science are generally weeded out. So be vigilant, keep your eyes on this as time goes on, because I think you'll hear about changes from time to time. And I want to recommend a book called Codebreaker by Walter Isaacson, which is really where I've got a lot of the materials for this. He traces the beginning of the CRISPR technology from its very start, and it goes into a lot more detail. And I highly recommend reading that, because it's he does a great job in that book. And I just wanted you to be aware of how this stuff works, if you hadn't figured it out before. And that, in my sense, and I'm probably a hopeless optimist, that the good from this new technology will be far more beneficial than the problems that it creates. So I think I'm going to stop there, and hopefully either learn something from this, or at least you had a chance to know about some things maybe you didn't know. Thank you.